And I will introduce you into advanced optical microscopy in biomedical sciences, and I will especially highlight challenges and potentials. Now, why do we need advanced optical microscopy? Now, if we, for example, want to optimize materials, and we have to understand how they are built up. In order to understand how they are built up, we really have to go to the molecular level. Really understand how is this material built up on the nanometer scale. And the same thing we have to do if we want to understand human diseases. We have to understand, not the body, of course, the body as whole, but we have to understand how cells, and especially how molecules interact on the molecular level. We are there also, again, at the nanometer level. Now, to understand cells and how molecules interact in cells, we need to study them live while they are doing something. And observation of living cells, we have to be non-invasive. And to be non-invasive, a good tool to do this is optical microscopy. Because optical microscopy, we can use light, and we can use far-field microscopy. Far-field means that we use focused light. So we can use lenses that don't touch the sample, they don't touch the cell, they are micrometers away, and we can also investigate, focus the light into the cells and investigate what's going on inside the cell. So that is far-field microscopy. On the other hand, we of course want to study specific molecules. And to study specific molecular processes, we somehow have to label them. That we know, okay, this is the molecule where we get the signal from. So we can do this with fluorescence. We can use fluorescent labels, either organic dyes or fluorescent proteins, and specifically label the proteins or lipids or molecules of interest. And then we can excite them with fluorescence light or with laser light and detect the emitted fluorescence from these molecules and look how this emitted fluorescence is distributed over space. And then we nicely see how the molecules, the labeled molecules, are distributed in space, like we see on the lower left corner for typical cell examples. So this is fluorescence microscopy. Now, fluorescence microscopy has been very, very successful over the years. It has brought us tremendous new insights into biological and cellular processes. We can go live cell, we can go in vivo, like you see with the fly there. We can go to endogenous molecules. We can look at how cells move, what you just saw with the movie. And that's why I'm working here in the Biomedical Institute, and I'm a physics person. And what we built up here is, we built up an imaging center. And this imaging center provides microscopes to users, especially to less experienced users in optical microscopy. And they, of course, come with all kinds of biomedical questions, and they have multiple demands. They have demands for temporal resolution, spatial resolution. Some want to look in 3 dimensions. Some need to do very deep inside samples, so we have to image deep inside samples. Some need to acquire data and images for over long periods of times. And of course, at the end, you get the images out, you have to do something with it, you have to analyze it. And most of the peoples, they need a bit of everything. And that's, of course, highly demanding. And I'd like to introduce you upon one of our projects, how difficult this is. So I'm in an immunological department here. And if you study immunology with microscopy, you have to study T cells, because T cells are the drivers of our immune systems. So if you have a target cell, like a tumor cell, that presents something from the tumor or from a virus to the outside world, the T cell comes in and recognizes these antigens, and it causes a close contact with this target cell. And once this close contact is formed, a so-called huge molecular rearrangements are happening, and the so-called immunological synapses forms. And when this synapse is formed, an immune response is triggered. Now, most importantly, we don't want this immune response to happen out of nowhere. So the T cell really has to recognize really specific viral or tumor specific things and not something else, because otherwise we have an autoimmune response. And so we have to study these molecular interactions to really understand what is going on there and to help curing our immune response. And the challenge is that these interactions that we are looking there, and I just circled them in a red square there, all these molecules we have to look at, that these interactions occur on different time scales from microseconds to minutes, hours, and on different spatial scales from nanometer to micrometer, because molecules have interact. When they interact, they diffuse. Diffusion is very fast, microseconds to milliseconds. And molecular interactions occur on the molecular level. That means on nanometers. But then in these T cells, you have early signaling, you have nanocluster formation, microcluster formation, and of course you have to look at the whole cell, how it forms. And this happens all on different spatial and temporal scales. So at best, we need a microscope that can cover all these different time and spatial scales. For example, where we were interested in is how are these molecular changes that you see in the T cell when it gets activated by antigens, how is it assisted by the actin cytoskeleton? And you might know that the actin cytoskeleton is just underneath the cellular plasma membrane. It's a cytoskeleton, kind of. But of course, it's closely linked to the plasma membrane and to different proteins in the plasma membrane. And therefore, of course, it modulates changes in the plasma membrane and therefore of these receptors that recognize the antigens a lot. So we wanted to look how this antigen, this actin cytoskeleton is changed over the activation. So what we did is we took T cells, we labeled the actin cytoskeleton or the membrane, and we put the T cells on a surface, and the surface was the microscope cover glass. So we came from below with our microscope, and we looked while the T cell was landing on the surface, and the surface was coated with antigen that were specifically activating the T cell. Now, the whole process takes minutes to hours. And the problem is optical microscopy because we excite fluorescence with a laser, and laser light can be toxic. So the irradiation can cause phototoxic effects. It can cause cell death or photobleaching of labels. So we moved to a so-called light sheet microscope. This light sheet microscope illuminates the cell only in a specific layer and then scans the layer through there. This is the so-called lattice light sheet microscope from Eric Betzig in the US. And he says this has a very low phototoxicity. So we checked that. Marco Fritsche from my group did that. And we checked this. And this is a movie that we took from a T cell just before it landed on the surface. And this is just the membrane dye. And if you look on the seconds up there, you see that you can really look at this over very long periods of time. So you really see, hey, you can do this for very long periods. Now, of course, we wanted to look at the actin cytoskeleton. So we looked at the actin cytoskeleton from the side and from the bottom, and you see green is a lot of light, blue is less light, and you nicely see how the overall organization of the actin cytoskeleton changes. If you look very closely again from the side and from the bottom, you see first it spreads, then it undulates, and after a while it contracts the whole cell, and the actin cytoskeleton changes. But of course we want to look at the cytoskeleton, and the cytoskeleton looks like our skeleton. It's like a meshwork or network. But if you look very closely on what we see with this light sheet microscope, and you see this in the upper right corner, the spatial resolution of our microscope is not good enough. It's 350 nanometers roughly, but it's not enough to resolve the network. But we have the so-called super resolution microscope, specifically a STED microscope, and I will introduce you to this later on. But this provides high resolution, below 60 nanometer spatial resolution in living cells. And you see this in the new image. The upper left corner is the conventional image, and the lower right corner is the super resolved image. And you nicely see now that we can resolve the actin cytoskeleton in the so far not activated T cell with very nice detail. And that's exactly what we want. And now we can take images of the actin cytoskeleton at different time points. The problem is we can so far not do it the whole time period, but we can take snapshot at different times. That's what we did. We took it after 60 seconds, 180 seconds, 900 seconds after contact. This is really at the contact area, the actin cytoskeleton that you see here, and you see massive changes. Now, I don't want to go so far in detail because this is not about immunology, but about the challenges of microscopy. So what we did is, because we have the image facility here, and we took different microscopes and interdisciplinary approaches to really look at this actin cytoskeleton with different detail, high temporal resolution, high spatial resolution, with multicolors, different aspects like calcium imaging and so on, contact areas and so on. And with all the information taken from these different microscopes that have specific different characteristics, we could come up with a complete knowledge about what the actin cytoskeleton is doing, so with more information. And that's exactly the message I want to take here. Optical microscopy. You have all these fantastic microscopes around, whether it's the STED-Microscope, a confocal system, a light sheet system. But you have these different demands like long acquisition time, 3D imaging, deep imaging, high spatial resolution, high temporal resolution. All these microscopes can touch some of these aspects, but there's no microscope yet that can cover them all. And that's why we need a lot of instruments to really cover all of these different demands. But of course, nobody can buy them all. So what we do here in Oxford, we share them. We build up facilities that can host a lot of these different microscopes, and they are fully serviced, managed, and we teach the people, and we discuss projects and advise the people what microscopes is best for their specific applications. But of course, we microscopists, we still want to develop microscopes that kind of can touch all of these demands. Now, of course, what you need also is chemistry, because you need bright labels and intelligent labels. But what I wanted to show you briefly, maybe some direction which microscope you could use. So the slide sheet microscope that I showed you, it has low phototoxicity, but it has a too low spatial resolution. Now, we went to Eric Betzig again, and he says he developed this enhanced structured illumination microscope. So instead of illuminating your cell with a continuous illumination, you illuminate it with a structured illumination, as I show there down there in the middle. So it's really a pattern with which you illuminate the sample. And you also use so-called turf illumination. So you illuminate really only the contact area at the cover slip, which for our previous application, to a part, was good enough. And if you do that and look, for example, on the actin cytoskeleton in T cells, you see this. You nicely see, you can image it for long periods of time, and you have nice spatial resolution to resolve really the actin cytoskeleton. Now, the problem is, because you could think, hey, we got the microscope that can do it. The problem here is that we can only image one layer, the bottom layer. We cannot go inside the actin cytoskeleton. And the resolution is still slightly limited. It is 100 nanometer resolution. But sometimes you need resolutions down to 60, 50 nanometers. So, what is the problem? Why do we have this spatial resolution image? So I told you, we work with far-field fluorescence microscopy because we want to be least invasive to the cells. So we use with focusing of light. So we are away from surfaces inside cells. Now, if we use work with focused light, like I show there in this sketch, you have the lens, the laser light coming in from the left, and you focus it to a small spot. The size of this spot is given, or this point spread function, how we say it in microscopy, is limited by the diffraction of light, and is given by this famous equation from Ernst Abbe. It's given by the wavelengths of light and the focusing strengths of the objective lens. So if we take visible light and the best objective lenses that are around, we roughly get to a lateral resolution of 200 nanometers. So our spot is 200 nanometer in lateral direction and roughly 600 nanometers in axial direction. Now, what does this say us? Well, if we, for example, scan this 200 nanometer large spot over a sample, we cannot distinguish alike objects at the same time that are closer together than these 200 nanometers. So any details below 200 nanometers appear blurred, like shown here. We cannot read small because it's blurred. We see a pretty blurred image here. The same here, for example. This is just a sketch, a very old cartoon of synapse vesicles. So our objects are these vesicles, and they are labeled. The membrane is labeled. And these vesicles are roughly 30 nanometers in size. Now, if we scan over there with our roughly 200 nanometer or 150 nanometer large spot, we cannot distinguish alike objects that are closer together than these 150 nanometers. So all of these vesicles appear blurred, like shown in this picture F. But if we make our spot smaller, because we raster scan the whole thing, if we make the spot smaller, we can distinguish alike objects. We can separate the signal from these objects in space and time. So if we make our spot smaller and smaller, what we suddenly see, we can distinguish the objects better and better. So this in scanning microscopy, and that's what we use here, the smaller the spot, sharper the image. So what do we have to do? We have to make our spot smaller. So how can we distinguish alike objects that are closer together than these 200 nanometers? Well, we cannot work a lot about the physics because this diffraction limited law, this wavelengths of light, we want to stay in the visible range. But we work with fluorescence, and we can turn fluorescence on and off. So imagine we could turn off all objects, but at a single point. Then this object, and then we scan through there, one after the other. We could distinguish alike objects that are closer together than 200 nanometers. Or we turn off all but a single objects, one after the other. Then, in all cases, we would end up with an observation area or a resolution that is much smaller than these 200 nanometers. Now, the upper part is turn off all but a single objects is the principle of Storm and Palm. But I want to go here to the principle of STED. So what do we have to do? We have to find a way how to specifically turn off the fluorescence in the outer periphery of our focal spot. How can we do this? So let's go back to our fluorescence microscope. We have the excitation light, the green, focusing into our samples. So we have a green spot of a roughly 200 nanometers in size. In this green spot, we excite all the molecules from their ground S0 state to their excited S1 state. And from the excited state, they decay back to their ground state, emitting fluorescence. But of course, the fluorescence comes all from this 200 nanometer area, so we cannot distinguish objects within there. Now we somehow have to switch this off. How can we switch this off? We come in with a red laser, red shifted laser, so in a fluorescence emission spectrum, really in the far red. When this hits the excited molecules, it forces them down to the ground state via stimulated emission, but we don't detect this. We only detect the spontaneous fluorescence, but the spontaneous fluorescence is not happening anymore because we force them down to the ground state by stimulated emission. So the degree of detected fluorescence, spontaneous fluorescence, is 100% without this red laser light, which we call the STED laser. But if we increase the intensity of the STED laser, we can switch or inhibit fluorescence, the normal fluorescence, more and more because we force more and more molecules down to the ground state without emitting fluorescence. Now, if we switch off the STED laser again, we are back to 100%. Now, so far, so good. We switched off all the fluorescence. We have a dark microscope. We have to do it in a controlled manner. We can do this by inserting a certain optical element into the STED laser that inhibits the fluorescence. And this optical element, when we focus it, distorts the beam in such a way that when we focus it, we end up with its donut-shaped form, which is in the upper right corner, where you see this red donut-shaped thing. And that's exactly what we need, because now the inhibition is only happening in the outer part. And now the nice thing is, by increasing the intensity of the STED laser, the area in which spontaneous fluorescence is still allowed, the central area, is getting smaller and smaller and smaller. And that's one unique feature, because we can tune the diameter of our scanning spot, the effective scanning spot, and thereby the spatial resolution of our microscope with the intensity of this added STED laser, this donut-shaped laser. And in this case, we can tune it from 200 nanometers down to, in this case, below 50 nanometer, but in principle, it's unlimited. Now, let me show you this with images. So this is a substrate diffraction. So this is a normal image, 200 nanometer spatial resolution of fluorescent beads aligned on the surface. And you see pretty blurred image. And if we take a line profile through some of these blurred spots, they are 200 nanometers in size. Now let's increase the intensity of the STED laser. If we increase it, two things become obvious. If we look on the left part, we see that some of the spots get resolved much better. And if we make a line profile through there, the size of these spots gets smaller. And if we increase it further and further and further, you nicely see that we can resolve the individual spots better. And the line profile goes down to 20 nanometer, which is exactly the size of the beads. So we really have molecular scale resolution. So, and that's why in our previous example, we can so nicely resolve the actin cytoskeleton of our T cell. And the nice thing, compared to the spatial, the pattern light microscope, the spatial structural illumination microscope, is that we now can do this in 3D. We can go inside the cell because we can focus. We can image several layers. But of course, we can also do dynamic imaging. And that's what I wanted to show you in the next example, my last example. What we can do is, we can look at the plasma membrane organization, because that's a very nice and important thing. Because if we look at the plasma membrane surrounding the cell, there is a lot of happening here, because a lot of cellular signaling events, receptors and so on, they get triggered in the plasma membrane, in the outer envelope. So especially the lipid and the envelope, the plasma membrane is a lot built up by lipids. And these lipids build this bilayer that you can see down there below. And the lipids, they have a head group that sticks out of the membrane, and a tail, these tails that go inside the bilayer. And they are involved in cellular signaling as well. And they interact with proteins as well. And especially what is important, it seems more and more important, is how are they packed? So if we have lipids, unsaturated lipids, you see in the lower left corner, that's an unsaturated lipid that doesn't have a double bond in their chain. So they are straight down. It's also in the upper right corner. And these straight down lipids, they can order themselves very, very tightly. So we call them liquid ordered environment. While unsaturated lipids that have, with the red circle, you can see that, they have a double bond in their chain. And this double bond introduces the kink, and that you see in the lower right part. This kink, of course, these lipids need more space, and that's why they cannot pack that much. That's why we have a disordered lipid phase. And this environment, whether a protein is in the ordered or disordered environment, makes it different. For example, these are molecular simulation. This is an immune receptor, CD2. And we just simulated what is the most preferable state of this protein, an estimation in the liquid disordered environment and ordered environment. You see that in the ordered environment, it sticks out a little bit more. And of course, if it sticks out more, the receptor is more accessible to anything in the outside world, and it can trigger much better. So the environment makes it a big difference. Now, if we talk about lipid environment, one thing that comes up immediately are lipid rafts or lipid nanodomains. These are so-called cholesterol and sphingolipid enriched small domains in the plasma membrane, very tightly molecular packed, so highly ordered. So a lot of saturated lipids in there. And certain proteins like to go in there and others don't. So that's why certain proteins go in there, do something together, and thereby maybe compartmentalize cellular processes. Now the problem is that these nanodomains are supposed to be very heterogeneous, highly dynamic, and extremely small. Smaller than 200 nanometers. And we heard 200 nanometers before. That's the resolution limit of our conventional microscope. That's why we thought we could go in there with our STED microscope, our super resolution STED microscope, and image these domains to have this missing temporal and spatial resolution. So we used two different lipids. We used the phospholipid and the sphingolipid. Now the sphingolipid, the sphingomyelin, is supposed to go into rafts. And the phospholipid, the phosphoglycerol-lipid, is supposed to not to go into rafts. Now, of course, to use it in our microscopy, we have to label it with a dye. So we labeled it at the chains with a dye R26479N. So we have a fluorescent lipid analogs of the phospholipid and of the sphingomyelin. I will call it PE and SN. So what did we do? We took our cells, live cells at physiological conditions, and took our labeled lipids and integrated them via a BSA complex very efficiently into the outer plasma membrane leaflet. So they are in there, fully incorporated. And then we took our microscope, whether it's the normal confocal or our STED microscope, and imaged the distribution of the lipids. And mainly what we saw, and you can see this in the lower left corner, is we mainly saw homogeneous distribution. So these bright spots that you see there, these are internalized vesicles. But the plasma membrane is very homogeneous. So instead of imaging it, we thought, because imaging takes a while, and as I told you, these are very transient, highly dynamic, and the imaging process, because we have to raster scan the cell membrane, takes a while. So all these heterogeneous are maybe gone. So instead of looking at specific hot images, we thought, let's look at the dynamics of these molecules. So we just parked our spot instead of scanning it, and then we waited for the molecules to diffuse through our observation spot. And that's the counts over time, you see in the lower left corner, look like this. So it's time on the x-axis and counts on the left axis. And what you see here is this is the signal over time, because we lowered the concentration of the fluorescent lipid analogs very much. So most of the time, we just see background noise. But from time to time, these lipids diffuse through our spot, and we see these huge bursts. And now, of course, we are interested in how long did it take the molecules to diffuse through our observation spot, because that tells us something about the mobility of these molecules and whether they are in domains or not. So we can use a technique called fluorescence correlation spectroscopy, where we just calculate the correlation function out of these temporal fluctuations. And this correlation function decays with a time that is characteristic for the process that is causing the fluctuations. Now, in our case, it's diffusion in and out of the observation spot causing the fluctuations. So the decay time of this correlation curve is roughly the average transit time of the molecules through our observation spot. And out of that, we can calculate molecular mobilities and molecular diffusion coefficients. Now, we are interested in domains. So we wanted to record FCS function at the same time at multiple spots. Now, we cannot raster scan the whole membrane, but what we can do, we can scan the spot very quickly with really 4 kilohertz or something like this across a line. And then when we scan it across this line multiple times. So for each pixel along this line, we get a fluctuation of the fluorescence over time. And out of that, we can calculate the correlation function. So we get a decaying FCS curve for each pixel along this line. And then we can plot it a little bit different, like now I show in the upper right corner. So it's space along the x-axis and mobility along the y-axis, and it's decaying from red to blue. And most important is the decay time. The decay is roughly where the yellowish area is. And if we have peaks to the bottom, then we know this is an area where we have slowed down diffusion. So we took our phospholipid DE and a sphingolipid SM again, and we recorded these mobility maps. This is the conventional recording, the confocal recording, 200 nanometer spatial resolution. And you see there are some peaks here. This recording took roughly 10 seconds. Yeah, there are some peaks, but it's not very pronounced. But if we go to the super resolve, the STED-Microscopy recordings, you nicely see that for the sphingolipid, for the phospholipid, it's pretty homogeneous. For the sphingolipid, we see these spots, these smaller than 80 nanometer large spots of slowed down diffusion. And they are transient in a few second time range, because if we record the next mobility map, the next 10 seconds, and so on, these hotspots change space and time and so on. So they are transient in a few second time range, and they are only visible in STED, because you hardly see them in the normal confocal recordings. What's going on there? So we have to discover the diffusion mode here. Now, the nice thing with our STED microscope, we can tune the diameter of our observation spot with the intensity of our STED laser. That's what we did. So we did STED FCS. So we recorded FCS data for different intensities of our STED laser, so for different sizes of our observation spot, and out of that determine the transit time. Then we calculate the apparent diffusion coefficient. And remember, large diffusion coefficient means fast diffusion, small diffusion coefficient means slow diffusion. And then we plot the dependency of this diffusion coefficient on the diameter of our observation spot. And this dependency varies for different diffusion modes. You can simulate that, what you would expect. So let me guide you through there. So you see here apparent diffusion coefficient in dependency of the observation spot diameter as expected for free diffusion. For free diffusion, constant, because no matter at which spatial scale you probe this, it should stay the same. But if we have trapping, so a molecule moves, interacts with something for a time, stays there for a while, is trapped, and then moves on, interacts something, moves on, we would expect that the apparent diffusion coefficient goes down for smaller observation spot diameters, so that the diffusion apparently slows down. Well, that's because the smaller observation spot, the more the transit is ruled by this trapping interaction. Now, what do we see? What do we see for our recordings? Well, for this phospholipid, we see very straight line, so free diffusion, only weak interactions. But for the sphingolipid, which we expect to go into rafts, and these rafts are kind of, these small domains are kind of trapping sites, we see that it's continuously going down. So we have trapping in this case. So the molecules move, interacts with something, move, interact with something. And as you see, the area, the slowed down diffusion relative to the phospholipid, we only see for very small observation spot diameters, so only for the STED recordings. And that's exactly what we see for our mobility maps, large diameters, small diameters for PE and SM, suddenly slowed down diffusion only shows up in the STED recordings, and that's why these hotspots, that's exactly where we have this transient trapping. And we can look what's going on here. What kind of trapping is this? We probably have a complex on the molecular scale with something, a protein, that is immobilized or very slow moving. This trapping occurs roughly 10 milliseconds. There's no movement during trapping. So it's really the lipid comes in, interacts with a protein that is trapped, or no, that is immobilized. The lipid stays there for 10 milliseconds, then moves on. This interaction is cholesterol assisted and actin mediated, because if we remove cholesterol or actin by drugs, we have free diffusion as well. And it's lipid specific. If we change anything on the structure of the lipid, we see that this trapping interaction is different. But if we change the label, because the label, of course, has an influence, so we have to do a lot of controls, and that's what we did. But we checked different labels at different positions, and we didn't see a dependency. So the current picture is that we have protein-lipid interactions. And maybe we have a formation or color sense into these transient hotspots. So the sites of the strong hindrance hotspots, they are smaller than 80 nanometer size, few seconds lifetime, constant exchange of lipids. Lipids move in, interact with them, move out for 10 milliseconds. Cholesterol assisted, actin dependent, and it's sphingolipid specific. So this brings me, so what might be the purpose of these hotspots? Well, bring molecules together, let them interact with each other, and thereby trigger cellular signaling. So this brings me to the acknowledgements. I have to thank a lot of people in my group, which is a mixture of different people from physics, biophysics, biochemistry, especially Sylvia, Marco, Erdinch, and Falk for the actin microscopy work and the membrane work. I have to thank the Imaging Center with Chris Lagerholm and Esther Garcia and Dominic Waith that are managing this whole thing. I have to thank all the people here in the Institute, and Al-Neelain, especially Simon Davis, with which we work a lot together. All the other microscopists in Oxford at Micron, like Martin Booth and Elan Davis, chemists, Harry Anderson, who helps us a lot. And of course, I have to thank a lot of people back in my time in Stefan Hell's lab, where we started all this lipid work, especially Alf Honigmann, and the chemists who synthesized all these lipids over the years, Vladimir Belov and Günter Schwarzmann. And I have to thank funding, the Wolfson Foundation, the MRC, the Radcliffe Department here in Oxford, and the Wellcome Trust. And thank you for your attention.
Thank you a lot, Professor Christian Eggeling from Oxford University, for this very valuable insight into super resolution microscopy. We're very pleased to have you today as a special speaker. I just want to ask you two questions before giving the chance to my colleagues. So regarding your second part of the presentation, so it is not possible to do two-color STED FCS to label simultaneously actin and the phospholipid.
That's what we do. That's what we do, exactly. That's what we do, because we want to now know, of course, what are these hotspots? What are the molecules behind these hotspots? So we counter-label other molecules and look whether these hotspots coincide with, I don't know, clathrin-coated lipids or other protein clusters or actin and so on. And that's what we're doing at the moment. That's exactly what we want to do. And of course, we want to also know whether, while molecules transiently are trapped, whether molecules move together. That you can also find out with dual color or fluorescence cross correlations.
Okay, cool. And also your image of T cell with the actin mesh, they're really cool. I just want to ask you, so under which physiological conditions, so which temperature you took those images?
We try to do everything at 37 degrees.
Okay, and cool.
5% CO2. So we have a normal incubator. So all the microscopes here, we try to equip them in such a way that we can really work with living cells. So 37 degrees and the right atmosphere so that they are alive as long as possible.
Amazing. And they're endogenously expressed, right? They're not transfected.
Some of them are transfected. So to get, for example, to get the actin labeled in the living cell, you have to transiently transfect certain, for example, live act citrine is a nice live cell marker for actin. But of course, we always try then also to have introduce controls where we fix the cells and label endogenous molecules, just to have a comparison whether we have any artifact from this expression. And of course, you have to work that you don't overexpress molecules. because that might introduce.
Okay, thank you a lot.
Yep.